Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about the life cycle of Aspergillus species. That means its morphology and sexual reproduction, asexual reproduction, how they produce their offspring or new individual. So let's discuss. At first, mycelium of Aspergillus or morphology. It is well developed and made up of loosely interwoven mass of hyaline, bright or pale colored, extensively branched septate hyphae. They have septa. Some of the hyphae ramify superficially upon the substratum. Most of the mycelium grow rapidly on the substratum, that means on where they grow. Other penetrate deeply into the substratum. Some hyphae grow deeply into the inside the body of the substratum. The latter, that means the hyphae, those grow inside the substratum body, absorb food for the entire mycelium, and the hyphae, those are freely branched and form dense mat on the substratum, help in reproduction. The slender delicate hyphae have thin smooth walls, cell walls. Next, reproduction. Asexual reproduction takes place by the formation of conidia. These are conidiophore. These are conidiospores which are formed on the conidiophore on a specialized cell called food cell. Okay, so asexual reproduction takes place by the help of conidiospores. Next, sexual reproduction is rare in nature. Majority of species of Aspergillus are homothelic. That means male and female sex organs are present on the same hyphae or same mycelium. However, few species are heterothallic. For example, Aspergillus heterothallicus. It takes place by the formation of in case of sexual reproduction takes place by formation of male and female sex organs. Male sex organs are known as antheridium. Female sex organ is called as ascogonium. Male branch, the total branch on which male sex organ is developed, is called as polynodium. Female sex organ is called ascogonium and the female branch is called ascicarp. Okay, next, ascogonium, ascicarp develop on the mycelium. Ascicarp means the female branch or the branch on which the female sex organ is developed. Archicarp on the mycelium in the form of septate loosely coiled structure. The young archicarp can be differentiated into three parts: basal multicellular or multinucleate stalk, middle unicellular and multinucleate ascogonium, apical unicellular multinucleate repetitive organ called trichogyne, and male sex organ is called as antheridium. Polynodium, that means male branch grows up beside the archicarp on the same or adjacent hyphae. It gets spirally coiled around the archicarp and arches over the apex of ascogonium. It can be differentiated into upper part and lower stalk. Next, fertilization. The tip of archegonium arches over the trichogyne and fuses with it. Actually, the tip of antheridium arches over the trichogyne, that means female part, and fuses with it. The wall of contact dissolves and thus making a continuous passage. Next is plasmogamy. The content of antheridium pass into the ascogonium. The pairing of male and female nuclei takes place inside the ascogonium. So antheridium calls over the female part of the mycelium and the point of contact then dissolves, and after the point of contact dissolves, the male and female nuclei pairs with each other, and then the pairing occurs inside the ascogonium. Next, the development of ascocarp. Whether antheridium is functional or not, the ascogonium in all cases develop into fruiting body called ascocarp. After pairing of male and female nuclei, ascogonium becomes septate. separate from other part of the mycelium. Each segment consists of one male and one female nucleus. Suppose it is the whole ascogonium, they get septate, and each part contains male and female nucleus like this. So this is generally dikaryophase containing male and female nucleus. From this dikaryotic segment arise ascogenous hyphae. Each ascogenous hyphae is multicellular with pair of nuclei and produces asci by crozier formation. What is crozier formation? Crozier formation means the terminal part of this binucleate cell of ascogenous hyphae elongate and form a hook-like structure, that is called crozier. Both nuclei divide in such a way that in such a way thin spindle apparatus are oriented parallelly in vertical direction. The penultimate binucleate cell acts as ascus mother cell. The nuclei of these cells fuse to form diploid nucleus. The diploid nucleus first divides meiotically to form 4 haploid nuclei. Each haploid nucleus then divide meiotically, and thus 8 haploid nuclei are formed, and this is mitotically, not meiotically, mitotically. Second division is mitosis, and from 4 haploid nuclei 8 haploid nuclei are formed finally. Next, each nucleus gets surrounded by cytoplasm and develop a wall. Thus 8 haploid ascospores are formed in each ascus. Cytoplasm left over in ascus is known as epiplasm. The asci may be globose or pear shaped. As the asci developed, a large number of sterile hyphae grow around them and form protective covering called peridium, and the entire structure is the fruiting body of Aspergillus known as ascocarp. And as this ascocarp has no opening, here in case of Aspergillus it is called as closed ascocarp or cleistothecium. It is the fruiting body of Aspergillus. Next, structure of ascospore. As the asci mature or asci mature, the ascospores set free by dissolution of wall or peridium. They are liberated after the decay of ascocarp wall. Each ascospore is fully wheel-shaped, unicellular, uninucleate, and attains a diameter of approximately 5 micrometer. And the wall of these ascospores are differentiated into 2 layers: the outer thick sculpturous epispore and inner endospore. After falling on a suitable substratum, each ascospore germinates to give rise a germ tube which develop into new haploid mycelium again. So it is all about the life cycle of Aspergillus. Okay, Aspergillus, first antheridium and ascogonium is developed, then plasmogamy. After plasmogamy, karyogamy takes place. Then karyogamy forms the asci young ascocarp, which undergoes meiosis to form ascospores. It is all about sexual reproduction, and asexual reproduction takes place through conidiospores or conidia, chain of conidia. So it is all about the life cycle of Aspergillus. I hope this video will be helpful to you. If you liked it, please hit the like button. Please share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.